Hello, everyone. Can you hear me? Okay, nice. Oh my. I think I work hard, but not just for set up the microphone. Okay. Sorry a lot because of the room is not open yet, so I could not come sooner for set up the computer. I think next time I need to install this software to my computer. It's more easy. So still hear the voice, right? Okay, thanks. I didn't hear your voice yet. So I need to copy the file to the. Wait a minute before I copy the file to. Okay, thank you. Okay. So I wish to see your face. It seemed to the first day we met. I mean, it's interaction, not just a post a video in the website and just a comment there. So yeah, thank you very much for joining me today. We have an hour already. So we kind of late because of my post, I think. Yeah, absolutely. And I think the midterm is in this matter kind of special, and I'm thinking about that. So at least I think is that I need to speed up something for transfer some kind of reflection to you. First, I think it both way. First, I will open the Zoom meeting every week, and also I post the video to you every week to we can catch up the schedule before the midterm. But about the and another one is I also post my PPT file for you can review what you need to learn. But actually, you need to the book is very necessary for the test. In the midterm is maybe is no problem to you, I think, because of with my experience in this course in 2 class I teach already. So we have quite a high score in the midterm. It's because of this technology in the midterm. It's quite similar with what you learn already. We just go a little bit deeper about the genetic. Also, we have a little bit of cell, protein, and biochemistry. Before midterm, I will review what you need to learn, and I didn't prepare the test yet. We had a multiple choice, but in this year maybe we have no multiple choice because of the limitation in the organize of the text. So about that, do you have any question to me? Okay. If no, so I will start the lecture today. So this one is the lecture I sent it to you. So this kind of I go fast. This one is like kind of briefly talking about biochemistry and also related with universe physics. And I just focus a little bit cells. You know, because of the wait a minute. Okay, so the voice is okay, right? Um, why I need to cover this one because of the some part of the task is a cover for this energetic part. So what is this? So bioenergetic is the study of the energy in the universe. But now we focus in the living cells, the basic unit of the living thing, and all of the chemical process underlying this the tradition. So why is that? Because of the cells, you know, is a very basic unit, it's a complex entity in the universe. Because they are a system in the universe, so the cells also follow the rule of the universe about energy. Right? So I think you still remember some simple rule about energy. So I just remind you some part. So what is the basic concept of the thermodynamic thing? So we had two rules, the very big rule about the thermodynamic thing. So first we need to consider the term is what is a system. So the portion of the universe which we are concerned. So we have a very big scale is our universe. I don't know we have another universe or not, but we still have we know it's universe, our current universe about 15 billion years ago they create. And also the surrounding environment of the system. So the universe, the system, maybe the galaxy or the solar system or our planet, also a very big system. And we have the system of our body, also a big system, and go to the more basic level, we have the cell system. So we have a system and the environment around the system, and also we have isolated system and a closed system and open system. So isolated system is we cannot change the matter. That means the material inside of the system the matter cannot go in or go out. And a closed system it change only the energy, An open system can exchange both material and also the energy. Energy may be the heat or some kind of the heat, yes, the heat, and some kind of maybe the light also. So this kind of this thing is quite a close. The bottle, hot water is kind of quite closed system. They cannot change energy, but they change energy a little bit, but the water in there cannot go outside, and another chemical from outside cannot go inside. So open system open system now we have to link to our cells, right? So we have a cell membrane, that is open system. Why is open? Because of energy can easy to go in and go out of the cells. And next thing is the cell need to drink, need to eat some chemical from outside, and also they expel some kind of chemical, maybe anastase of the biochemical pathway in the cells. So our cells is an open system. So this is a visualize for that kind of thing. So this open here. We have no the cap, in cold we have a cap, and isolate system. In the world right now, we don't know is which one is a completely absolute isolated system because of the temperature, the energy, the heat from the inside always released to the outside. So our planet is a isolated system or the closed system, because you see that there's nothing can get out of the earth because of the force in the earth is recruit all of the material still in the earth, right? Our earth is quite I think closed system because of the energy from the sunlight can get in, and our earth also reflects some kind of light to another part of the universe. So our planet is a closed system. So another also quite a basic thing is enthalpy, the heat of the heat content of the system. So it reflects the number kind of chemical bond in the reactant and product. It's all the heat in there. So it means this also is not the heat yet, but this is the chemicals. Is store some chemical bond in there and easy to expose the heat if we can find the paper. So delta is just measure the before and the after change of heat of the system. So we have exothermic and endothermic. So it just remind you something. So delta less than 0, I mean the heat is lost out of the system. Another one is quite entropy. That's been the quantitative measure of the disorder of the system. I mean, if for example, in our classroom, if the table and the chair is kind of in order, it's very beautiful. So that means that is order, well ordered. So this way we have earthquake. So we have some kind of mixing and messy in here. So the chair and the table is go different place from the order in the beginning. So that is entropy. The disorder amounts increase in the system. So we have moved to the rules of the thermodynamic rule. So we have to admit that all biological reaction and process follow the law of the thermodynamic. So we have the two rules. What is this? The first law of the rule is total energy of the universe must be constant. I mean, so if the system is constant, if the system is isolated, all the energy in there should be not changed. So energy may be converted from one form to another and move into another system from this material to another material, or the energy can use to do the work, but it cannot be destroyed or created in any kind of circumstance. Very basic one, right? So And the second law of the thermodynamics thing is in all nature process. So the entropy of the universe increase. So the entropy of the universe always increase. They not decrease. So we had a question like living system is highly complex and organized. You see that, right? The cells, we have the cell membrane, we have the Golgis, and anything is from very complex, from very complex monomer, they reassembling to the polymer. I mean, the order is now a highly perfectly organized. I mean, the entropy of within the cells, they should be increased. No, sorry, the entropy should be decreased because the order is followed very good. So do they violate the second law of the thermodynamic or not? So here, the figure illustrates for the answer. So you say that this is a monomer in the cells. It's a monomer in the cells. And it looks like the cell has a cell membrane. They isolate them from the environment. And then when they involve, when they living, they make the polymer from monomer. But the problem is outside is also the same thing. They also have the connection with the outside. So in that process, the cell is using a lot of energy, and they produce the energy. Now they store in the bond of the chemical, the polymer in the cells. But they also release a lot of heat outside in the environment, surrounding environment, and making molecules outside of the cells. They move very fast. They increase the entropy. So any kind of this is after analyze this kind of thing, we see that the cell also very strictly follow the thermodynamic rule of the cells. The second rule, the entropy should be increased. Another term I need to mention is free energy. This one is very basic for a kind of for the cell can work. For any kind of part of the cell, molecules of the cell can follow that rule. So free energy is the amount of energy capable, that's useful energy to do the work during the reaction. So we have the useful work and useless energy. The useful energy and another one is the useless energy. So we have the two kind of reaction related with free energy. So exergonic reaction, so they release energy. So from if we have the substrate, the reactant in here, and this is the product. So if the product had more energy, so that means this is a endergonic because of the energy in here from the left side, they have very less energy, and now they some energy put in this one. So the product is now have more energy. So we have the delta G is less than 0. So this is endergonic. So they need energy for the reaction. Exergonics is the reverse way. So from the high energy reactant, they change to the low energy product. So you don't need to remember this one. So delta G relate with the enthalpy, and this is temperature, and this is entropy. And delta G we can measure using some reaction. We can measure any chemical in there. Now I heard the door here. So can you turn off your microphone a little bit? Okay. This is illustrate for the exergonic or endergonic reaction. This is stand for reaction. This normally we use in the chemical or the enzyme experiment. So in here, the reactant, they release energy, become the product. So this one is exergonic reaction. So the energy we release from this reaction, and this type of reaction is easy to happen, right? Easy to happen because of this is quite important. This is, for example, this is a chemical, and they store strong energy in there, and they kind of is high energy level and tend to have a lower energy level. So if you work in a protein field, you really focus on this one because the protein is sometimes it's unstable. And if you want to make this protein stable, you have to release internal energy of the protein. So the protein now structurally becomes stable. And another one is an endergonic reaction. The reactant is absorbed energy. The product is in the end state, they have more energy than reaction. So this reaction is need energy put in there to happen. So I have a question. So for example, this one. So if the reactant can change to the product and release the energy. So if we want to turn product to become the reactant, we can do that, right? But how about energy in here? So energy, just we put this amount of energy back to this reaction to make the product become the reactant or not. We can change the product to the reactant. Oh, I have a question. Oh. Oh. So anyone still remember? I cannot see the chatting. So okay, very nice discussion. Thank you. Yeah. We don't need a lot of energy. but we need the energy. This energy should be this energy put in the reverse way should be the more the stronger, larger amounts compared with this energy. That is the rule. It's not if this one is same. So from product to reactant reverse way, if the same energy, so it's not happen. If that one is we have the absolute isolated reaction, isolated system. So we have to put more energy in that. So you have to remember this one if you work onto protein, onto protein optimization. So if you work on protein optimization, if we have to put more protein, sometimes you need to put some heat into the reaction or some kind of more product in there. Now the product becomes the reactant, and this reactant becomes the product, right? Okay. So we talk about the feasible of the reaction can happen. So for example, it's the man from the high state, so he easy to jump to the river, jump down to the river. So that is happen very easy, right? But from the river, he can turn back in here quite hard, and he needs a lot of energy to come back. And another one is also very important in the cell activity. That's the concentration. The chemical from the low, from a high concentration try to become the low concentration. That's also one type of energy, and you see later. So if you have a membrane like this, and you have a membrane like this, and this is high concentration chemical, is low concentration chemical, and high concentration chemical is trying to turn to the plate with a low concentration. And that's it moving, it will make the energy. And you will see clearly in the, maybe in after midterm thing. And a chemical reaction, there's one rule, not always accurate, but I think it's almost accurate in many kind of reaction. So the polymer, they store the energy in there. The polymer always high energy than monomer. If you want to make the polymer from monomer, you need to put a lot of energy. And if you want to have energy, you just break. You need to just break the polymer to become the monomer. So like glucose, it has a 6C, and now you cut it, become a 2C, like pyruvate. So you will have the ATP, right? That is the process of the cells with the help from the enzyme. So the spontaneous processes is the easier from the high concentration, high energy to the low energy state. And but that is not always correct. They have something we need to explore. That's we have the barrier of the chemical reaction, also the biochemical reaction. So what is the barrier? So you see that here, if this man, he tried to jump from here to here, he need to work a little bit to jump, right? That's quite easy. And it's the same thing for the paper, the seed in here. If this one is store a lot of energy in here, it becomes a fire. But they cannot automatically, spontaneously to become the fire, right? I need to fire it a little bit, lit it a little bit here, and then they can activate it, and this become the fire and release a lot of energy. So we have some activation energy. Activation energy provide energy for reaction state from the high state to overcome the barrier to go down to the reaction from the high energy state to the low energy state. So the enzyme, you know, is enzyme is trying to reduce this barrier reaction from the high to the low. So the reaction is easy to happen. So like this is a function of enzyme. So enzyme will reduce the bar, and the reaction can easy to work. Yeah, this one. So I can quite fast through this one a little bit, because in the second, because after this I will show you about protein chapter, and very closely, very hard, very clear about this one. But this one is, maybe we stop here a little bit. Transition state is what? You know, is in the chemical reaction. So the two reactants move randomly inside of the tube, right? For example, in the tube. So they slowly moving, maybe at the low temperature, maybe at the low temperature, they kind of moving slowly, and they not touch each other, and the reaction not happen, right? But in the cell, also the same thing. We have the cells, and we have a lot of chemical in there, billion kind of chemical, and then they not match each other because some chemical is kind of low concentration. In here is they not match each other. Enzyme will provide them the room. For example, this is a active site enzyme. So enzyme is just accept for this chemical and another chemical, like because of they was born for each other. So they fit each other, and two chemical is come easily and not interfered by another chemical. So the reaction happen. So enzyme is one of the function of enzyme is they provide the accommodation position for the chemical, for the protein or another chemical can enter, and then reaction happen in there. So that's the one role of enzyme. So enzyme in the cells is quite different with in the chemical reaction. You know, it's chemical reaction. We have the A, B, C, maybe just very few chemical in the chemical reaction. I mean, it's like the inorganic or organic reaction in the chemistry. But in the biochemistry, in the cells, that thing is not happen because of from the subject one. Enzyme will be modified, become the subject two, and the subject two, they cannot back to the subject one, even though the equilibrium is maybe the concentration of the sub 1 and 2 is kind of equal, and then they stop. Normally in the chemistry, but by biochemical reaction, so subject 2 quickly will catalyzed by the enzyme B. It change to the subject 3 and subject 4 and 5 with a different separate enzyme. So that is and the concentration of the substrate is really fast use, and that is we call it biochemical pathway, and quite some reaction very fast and some reaction is very low. They affect by some factor. Here is enzyme, you know, right? So enzyme is we have the surface A's in here. So enzyme, you may also know it already. So they speed up the reaction rate, and they can reuse when they disassociate with the product, and then revert to catalyze for the next round of the reaction. So enzyme is very effective from the nature to us. And this picture shows what happened in the cell. So maybe in the animation of the cell, you see some kind of cell organelles, the nucleus and Golgi or something. And between them is you think it's water, but actually not water. All of the cytosol in there is a chemical. The concentration of the chemical in there is kind of different. So you see dance of the chemical inside of the cells. It's different with a little bit water aqueous paste inside outside of the cell. So the chemical in here is very complex, and then they affect by a lot of the factor, like where is this? Like the concentration of the reactant, concentration of product, and the temperature of the reaction, and also the concentration of the enzyme. And back to the concentration of enzyme, you have what? you have the DNA and RNA activation. So I mean, the protein expression to make the protein enzyme concentration in the cell is changed. The reaction inside of the biochemical reaction is quite a they couple together. What is a couple? So like the reaction want to happen, but the energy in there is not favored because of the from the low state to the up state of the energy. So the energy is not favored, and that reaction did not happen. So enzyme is just reduce the barrier of the reaction, but they cannot provide the energy for the cells. So they cannot provide the energy for the reaction from the low to the high. So they have another parallel reaction to make the reaction happen. So they need to have another reaction. That the reaction will provide the energy for the second reaction. That's we call it couple reaction. So it's this figure to illustrate for this one. For example, we need the Y chemical to become the X chemical. We need to up the energy is now go up, and the delta G is now less than 0, and this favor red color is not favored, and this one is completely cannot happen. But in now with the enzyme help or with some enzyme help, so we have another reaction. So in here the C chemical become D chemical, and this energy from C released from C to D we provide to X Y go to X. And the delta G of this reaction should be larger delta G from X to the Y to the X. So that's it true. If they equal, they also cannot happen. So C to D, normally we use what? We use ATP in the cells, ATP in the cell. But ATP is not only one chemical provide the energy in the cell. We have another types of the energy. Chemical. Okay. So in here is obviously a postphase. We have explanation here. So postphase is different with the carbon. So somehow you see carbon is the atom is a structure of the chemical in our plant because of the last video I showed already. So right, the clouds of the carbon is 4. They need 4 more, or they can lose 4 electrons to become the stable. So postphase is have 5 electrons in the cloud. So in the layer. So they also very active atom. So somehow our planet, our living system using postphase for transfer the energy. So in the next presentation, I will show why is the postphase can change the protein or enzyme activity a lot. So in here you see that here, focus in here. So glucose and fructose will connect together in the reaction to make the sucrose. So delta G now is larger than 0, 5.5 kilocoulomb. So this one cannot occur. This is not favored. So in that case, glucose need to bind with ATP first here by some enzyme, not only by themselves, by some enzyme. And then the glucose can now connect with the fructose, also with some enzyme. And now they release the postphase and become the sucrose. And now if delta G is less than 0, and this couple reaction can be happen, of course, with the help from the enzyme. any kind of the reaction, our cell is need enzyme. And that is the main thing in our course. This one is, I mentioned already, the concentration reactant also is effect to the cell reaction. Another one is equilibrium of the chemical. So you see that we had a two direction from the left to the side and from the right to the left. So due to the concentration, when the concentration of B in the ratio, they're not equal, right? They're not equal in the rate, some ratio due to the reaction and also due to the concentration of enzyme. So B can return to the A, and that is stop the reaction stop. But B is normally they change to C or D or some another chemical. So this is not really exact. Okay. Next one is the activate carrier. So these are the molecules. They can bring the energy to the reaction. From this reaction, this is a couple reaction. This is a provide donor energy reaction, and this is a receive reaction for happen. So we need some carrier energy carrier. So the most you molecules in this case in our cells is what? ATP, right? So you know is what another molecules is also energy carrier in the cells. Do you remember some? Energy carrier. Yes, NADH. GTP, very correct. GTP also is guanine, right? Guanine. They can change to the phosphate to the. Okay, excellent. So here is a table of the chemical related with the. Now when they release phosphate, they will provide a lot of energy. Very a lot. Where is the energy? Where is ATP? Here, this ATP. This is the amount of energy when one molecule is released. 1 mole, you see, 1 mole of the ATP release. So ATP is the most used chemical in the cells and also in our planet, in the living thing. So another side information is if you want to detect something living in your sample. For example, if you want to detect some contamination, maybe the bacteria contamination, you don't need to detect the DNA or RNA or some kind of specific molecules in here. You should need to detect the ATP in your material, and you can have a conclusion. There's some living thing in here already. So that means this sample we have contamination. That is one thing, because only living thing can produce ATP for living. And you know, is in one day, our body can produce about more than 70 kilograms of ATP. 70 kilograms of ATP. But when they create, all of them is quickly transferred to the ADP for energy. So that is a lot of reaction happening in our cells, our body. So this one just illustrates for the energy release in the cells. So from the very high states of energy, when, for example, this one, if the paper seed is the fire, they will become the heat, become the fire and heat. The heat really will destroy the structure of the cell or destroy the room also. But in the biochemical, because of they are very flexible, they are very sensitive with the heat. They cannot allow for the heat can release a lot because of the cell will die, right? So a lot of reaction happen to transfer the energy gradually high, low, lower a little bit, and become the lowest state energy. So we have a lot of enzyme to make that kind of thing. So if you, for example, glucose store a lot of energy. So from the glucose, so the CO2, we have a lot of reaction, and each reaction will release a little bit of the energy, and the cell will save and use very effective energy inside of the glucose. And another one is maybe skip fast because of this just show up where is the energy for our planet living thing. So we absolutely accept this. It is the sun, right? Without the sun, we have nothing in our planet, and the rock is still the rock. And we have the photosynthesis is very good, very vital roles in our planet because they make sugar, and all of ours is heterotroph species. We can use sugar in the plant and you in the food chain. So in the final term I want to mention, just remind about the metabolism. So the food, we have two opposite pathway makes up the metabolism. Metabolism is including two kind of reaction. First, we have the catabolism. In the process, we break the complex chemical and we release energy. And next, we have the opposite way. We have anabolism. So that's it. We build the complex chemical, and again we store the energy in that complex chemical polymer from the monomer, from the simple form. So in here we have this illustrate for that. So it's a food, and we catabolism happen, and we have a simple form like the nucleotide, like amino acids, and we use that one, and we put some energy from ATP because this one is making ATP and another form of energy. And we use that information thing to make up a molecule for the cells, the structure, and useful for the work of the cells. And okay, so we have a catab and anabolic fat way, and some fat way is we call it anabolic fat way because they stand for both direction. For example, so acetyl-CoA in here we have a glucose here, so they make the acetyl-CoA from here, and from citrate. Oh, sorry. For example, we have a pyruvate and and glucose, and it turn to acetyl-CoA. This is catabolism, right? From here, they join to another reaction to make another chemical, like fatty acids and another amino acids. And from this kind of station, so we call this anabolic fat way. Okay, so we have a little break, and in the side, if you have questions, just forward to me so we can discuss. I'm not sure I can answer all the question from you. Absolutely. And we break about 7 minutes due to my clock. So, and then we turn to the next and very important chapter is talking about the protein. Okay, thank you. Have some coffee. Hello. So anyone is okay in home, right? Not in the coffee shop and chatting, right? Hello, anyone come back? Hello. Thank you. It's feel very lonely in here. Just a chair and a table in here. Just only me. It's quite a very terrible situation in our our school and also our society. Okay, so hope you very well in home or your room. Um, I also very hard to work at home, so I have to come to the university, and I also have some experiment in the lab. So because of all my student is go back home uh, uh, for avoid the pandemic, so only me in the lab. So okay, so we have just around 1 hour. Remember, thing. So I need to quickly move to the second part in our second presentation for our thing. This one is we start to talking about the real material of the cells. And chapter 4 is the protein structure. I'm sorry. And the function. Actually, the title is not maybe not very like the accuracy is not very high about the protein. In here we talk a lot of things about the protein, and the protein science still quite a very mystery in our human our scientists. You know, it's a lot of structure, a lot of protein structure is still is not published yet. So and a lot of protein we cannot synthesize by ourselves. Um, and a lot of things, a lot of the mechanism in the protein science also we don't know. But human and the scientists also go very a long way already to explore for the protein structure and the function, and also organization of protein. And from the time we know about the DNA, we know about the coding of the DNA to the translate to the protein is more and more interesting. It looks like our life is the coding, and the protein is a molecule to do the work from the code. And they need energy from the last chapter. So in here we have some topic to talk. Uh, sure. I upload all of my PPT to the Blackboard for you. What are my experiment I do? So I do in genetic and some experiment like a diagnostic. I also teaching a diagnostic and relate with medical genetics. So I also do in the animal, I do in human, also do the plant. And we have some a small project because I just come back from Korea just 1 year. So I have a lot of things to do. And okay, sure. I upload not only the video, I also upload the PPT file. So my experiment is kind of interesting, and I have quite a how much 18 students right now, but they own in home. Okay. So the content today we had 4 parts. I hope we have enough time for this, and some part I assume that you already know, so I can fact through is a little bit fast. So the shape and structure of protein, and how to protein work, and how protein are control, and protein study, how they study protein in kind of short but kind of necessary to you to know, explore the thing in the protein science and also the cell science. And first we need to discover, actually we just explore it. People is discover for you, ready? So the shape and structure of protein. So this one is, I think you all know about this. This is, we have the subunit of the protein. So the protein is, at the first, the scientists is really, they confused. What is a protein? What is protein? Because in that time, they don't know about the DNA, they don't know about the thing. They don't know a lot of things. They just, protein is something, they know about protein, but they don't know, they have no any tool to explore what is protein. And now is you all know is protein is a string. The sequence is a polymer. I mean, it's only one string of the subunit. Monomer is the amino acids, right? So and how about the you know, is in the sugar or the starch, you have the chains of the sugar, glucose, and become the branching, right? The branching. And the protein is different, and DNA also different. So protein, we have only one string. This one, and we have the next one, and the next one connects to the previous one. So this is surprise about protein is just only one string in the protein structure. And more than that is the scientists also find out the chemical formula of the major types of the amino acids. Right now in human, we have about 20, no, not 20. Actually, right now we have 21 amino acids in our cells. We have the number 21. So I will show that kind of picture for you. Is later we have the 21 amino acids, not 20. And it's a peptide bond. We have a peptide bond here. And firstly, is the structure of the amino acids. Structure of amino acids is quite simple, and we had a rule in that. So this one is we have amino group, and this is we have the carboxyl group. And this is a non-ionic form. But actually, in the water, the amino acids in the ionic form. So that means is one proton is they absorb from the water, right? And this is H in here. They lost one electron from the C terminal. So that is in the aqueous, in the water. So this is structure of the real structure of the amino acids. So this is easy to join to the reaction. So this is statement for any kind of some paragraph, some section of the book. You see that very easy to grab the thing in the book, right? So they statement that the shape of the protein is specific by its amino acid sequence. So next slide will show that. So first we need to know about, we need to remember about the peptides using their I forgot the reaction. So expels one water molecules from this, and we got a peptide bond between the two amino acids in here. So very easy to remember, right? So next, because of this, we had 4 amino acids here, methionine, and we had aspartic acids, leucine, and tyrosine. And for example, in the coding reason, methionine is an ATG, right? Methionine is the very first amino acids in the protein sequence. But you have to, this is information that the methionine is normally, after the synthesized in protein, methionine normally we will cut out the protein sequence. And for reading the protein sequence, we read from the left side to the right side, the same way with the DNA sequence. The left side we have the N terminal, and the right side we have the C terminal, like the N here, amine group, and this is a carboxyl group. So from the N to the C. So you have to familiar with this kind of way to call protein sequence. And some modification of protein, they also things like that. For example, before I need to synthesize some kind of peptide for vaccine development, because a peptide is a vaccine, right? So I need to use some cell system, and I need to call some company, and they help me to synthesize a peptide. But in a peptide, I need to modify something. So I want to add some kind of chemical in the N terminal. They ask me, do you want to add some kind of atom or molecules in the protein? So I want the yes. So which terminal one you want to add, or you want to add in the middle? So I want to add in the C terminal or N terminal. This is something related with biotechnology. And another one is also very important and very basic, and also useful for you for use that kind of thing for experiment is. You see that this is strain of the protein peptide sequence, and this is a N terminal and a C terminal, and this is a peptide backbone by the peptide bond, right? And you see this, it's color in here green and a blue light blue color in here. This one light green color or yellow in here. This is the side chains of the amino acids. And you know, is all of the chemical reaction of the protein or enzyme is or the structure of the protein. It depends on the organization of the side chains of amino acids. And why? You see that the N terminal and C terminal of each amino acid is similar with each other, right? But the only difference in the side chain, like the DNA, they have the base. The base is same, and sugar phosphate is same. And in here side chain, they based on the side chain, based on the side chain part, we have to divide 21 amino acids become the many group. So in here we have the negative charge group, positive charge group, or uncharged group, and a non-polar charge group. And in here is non-polar a lot. So we have a lot of things else. So I find out and show it. This one is not belong to our book. So we have this one is amino acids with electric negative charge, positive charge, and this is a polar or unpolar. And this is a hydrophobic side chain or hydrophilic side chain. That's very important for the protein structure organization. And in some special case, we have the aroma. This is aroma, the smelling aroma amino acids. Or we have the cysteine amino acids. Cysteine is very important amino acids for the protein structure because they have sulfur. Sulfur can make the covalent bond. You know, a covalent bond is the most strongest bond in the chemical, right? So sulfur in here will provide the covalent bond for a protein and make the protein is more very stable. So if you found a protein, it's a lot of the cysteine in the protein sequence. Your protein is very, very stable. And if you heat, you need to more heat to break the protein structure. And this is a selenocysteine. So sulfoin here is changed to the selen. This is amino acid number 21 in our list right now. The scientists identified this one is new one. And another one is interesting is for glycine. Glycine is the smallest amino acids in our list because see, it's very simple formula. I had one publication, and I study about the binding site of the protein. I study a binding site of protein, and you know it's the binding site like this, like our hand holds something, this for reaction. So I have the hypothesis that another scientist also have another hypothesis, but because of this negative charge is a positive charge and it bind well. That's the one related with the charge of the ligand and receptor. But my hypothesis is this one is because it's a small or big accommodation volume in here. So they also affect the ability of the binding ligand. Bigger have ability to bind more types of the ligand into their binding position. And in kind of my study, I found a glycine in that position. So glycine, because it's quite small, so my volume for accommodation for this one will be bigger because it's small, right? Something big, and this one will be smaller. So I try to focus this one in this year. So it's quite very interesting if you work in protein, and also very tough if you work in protein structure science. And another question, so what types of the bond, chemical bond? You know, is we have 4 types of chemical bond: covalent bond, we have the ionic bond, we have the van der Waals, or a lot of things in the water soluble thing is a hydrogen bond. So the statement is all of the type of bond is a contribute for the protein structure. Anything is important, especially in hydrogen bond because the protein in the cell now is all of them is inside the water, right? So we have in the water, especially in the water, ionic bond is quite very weak, and the hydrogen bond is also very weak. So but a lot of bond weak. Some of them weak bond become strong in the protein structure. And also very important post recently also published newer knowledge compared with this book is the scientists found that what's this? Hydrophobic and hydrophilic bond also very important for the protein structure, because you know is I'll show it. For example, in DNA structure, maybe you don't know yet. For DNA structure, you know it's complementary by A linked with T and C linked with G. Hydrogen bond is a main bond for DNA structure, right? But actually now it's time to identify another bond is more important. That is a hydrophobic thing. You know, it's this is for example, my hand in here. I hope anyone can see the video a little bit. For example, the sugar and the phosphate backbone is a kind of parallel for the amino acid for DNA, right? And then the base, the adenine, guanine, or cytosine, they are hydrophobic. The base is hydrophobic, and the phosphates and sugar is hydrophilic. A hydrophilic part will be turned out to touch the water in the medium, in the environment. But the base ATGC, they hide themselves from the water and they turn inside and they attack each other by hydrophobic force and also the hydrogen force. So we saw we more clear, more explore about the stabilize of the structure of the DNA. And the protein also the same thing. The side chains of the protein also is hydrophobic and hydrophilic. Hydrophilic will be turned outside to touch the water, and a hydrophobic will be turned inside to hide themselves from the water. And that also is an interaction with another bond, like hydrogen bond, ionic bond, and van der Waals bond. And sometimes it's called covalent bond to make the protein structure. It's a very complex process. And the complex process is if you have the short peptide, it's kind of easy to make the structure because of very weak. But if you very long peptide, and then they need to turn many kind of process to become the final structure, the stable structure. And sometimes it's not, if folding is not correct, and become the non-functional. Okay, this one I explained already. So this is the backbone, and this is the side chains. The side chain will be conformation and turn out with the hydrophilic outside and hydrophobic inside. So they make their structural protein. And this is also showing the importance of the hydrogen bond between amino acids. This part one amino acid, this one another amino acid, and you see that they contact very complex between this amino acid to another amino acid, and some kind of atom here. This atom they contact with two atoms in the next of the binding. So we have the backbone to backbone, we have the backbone to side chain, and the side chain to the side chains. A lot of bond and complex to make a protein structure. And this is really trouble for the protein scientists. No one can predict. No one can predict. Right now in DNA, because in DNA we have just ACTG. That's quite easy to predict. So now you just put your DNA sequence inside of the software. The software can predict for you the structure of the DNA structure, and in a single strand, I mean. But if you put the sequence of the protein into the software cannot do that. If you have the template, I will explain a template later. But if you have that's a new protein, new sequence, the software completely cannot predict for you the structure of the protein. If you can do that, you're very famous and become a Nobel laureate. Okay, now we come to very basic rule after we want to explore about the protein structure. Anyone want to explore a protein structure? Because if you know the protein structure, you can predict for the active site. You can predict for the target product. No, the target substance subject. And you can predict for the drug because the drug is interact with the protein to do something in our cells. And now is how protein folding. So we have the very simple but very right and acceptable for all of the scientists in the world. I mean, protein fall into the conformation of lowest energy. For example, in here we have the high energy with unfolding, and that means we have no any kind of contact between the amino acids. But when you reduce the energy, and the protein will automatically they form they form the bond between the amino acids. And, and that kind of stage in here is the lowest energy. Lowest energy. Very simple to imagine that kind of situation. If you want to explore that, you just simply heat the protein mix. If you heat them, and then they will be unfold, right? And then become their degraded protein. So why is the lower energy is a final form of for native states of the protein? So you know, it's a spontaneous leap from the high energy to the low energy. So the reaction or the energy for the system, now we consider protein is a one system, and the energy we release and return from the high energy state to the low energy state, it becomes a native form. This side of the what assumption. This assumption is used for some kind of computer software. You know, its computer software can predict a protein. They cannot predict totally, but if you provide the software some data, and they can predict it based on they minimize the energy between the amino acids or between the atom in the protein structure. I will show you in the end of the presentation about the method. I mean, it's briefly method. In our department, in our college, biotechnology, we have the Dr. Lee or Dr. Miss Joe is predict for this one using software. I'm not so specific in this one. I can produce a protein. So in here is the statement that the nature protein. I mean, the nature is unfolding protein. They can automatically recover their nature state. But this statement, like for example in here, so this is one protein. They form correctly in the cell already. So if you use some urea is normally break the bond inside of the protein, break the bond. And then because the bond is break, so the protein in nature is become the 3D structure is lost and become a string like this. And if you remove the urea concentration, they will slowly form the native structure. But this one is not always correct because if the protein is simple, maybe very short, or the structure is simple, they can do like this. But if the protein is very complex, never you have this kind of success. So my lab before is also working in protein production, and because of our protein is kind of very short, about 30 or 50 amino acids. So we can form like this. We can do this method, or you can use a heat, but heat is quite dangerous because heat will be make a protein after they denature, and then we will tangle with another protein, and they become messy, and never they can return the native form of the structure. Like when you heat the egg white, and they become a white color, right? And we have the bone egg. Another one is in the cell's activity after the protein they make from the mRNA, mRNA coding translate to the protein, and we have some another protein, small protein can help this protein is correctly formed protein. That's the truth. Without the chaperone, so the protein incorrectly formed is normal. So this one is applied a lot in the biotechnology because what? Because of you want to express a lot of protein in the cells, right? Because our cell is very powerful, they can make a lot of protein you want. And that protein is quite offensive if you are doing recombinant experiment using animal cells or human cells. And also is the yield is kind of low because of the animal cells. Normally they have the 1 day, they have 1 day or 2 day to doubling, right? You mean the cell mass is kind of low. But if you can use the bacteria with the 20 minutes for doubling, 20 minutes. So that means after 1 day, you have a lot of bacteria and you also have a lot of protein. But the problem with bacteria is the folding of protein, because of bacteria are very simple. They lack many kind of protein need for folding of the protein. So in the one of the application of this thing in the biotechnology, so we have to put some chaperone gene inside the bacteria and help us. And when the chaperone appear in the E. coli or in bacteria, so they have our target protein will be folding correct, and then they become they have function. Without a chaperone, they have no function. But that I tried before. I did it, but my experiment failed. It's not always successful this one because the protein folding is kind of very complex, and the scientist also is not sure about the successes or not. Success rate also very low right now. Interesting, right? If you're working on protein production. And another chaperone is kind of big, so they light a chamber, and for protein new synthesized can get in and become folding correctly inside the chaperone. This is okay. Okay, this one is and modifies it a little bit. Okay, any question about the part I share? Any question? You still there? Hello? No. Question? Okay, maybe you had another question later. So just tell me and we can discuss, because I also have a lot of questions. A lot of questions related to cells. Next thing is, you know, is the protein is very diverse. Diverse not only in the sequence and also diverse in the shape, right? In the cells, we have a lot of protein. I don't remember exactly, but we have about 25,000 genes. 25,000 genes. 25 or now scientists identify more than 100,000 genes. Even their regulation gene, not only the structure gene like making a protein. They just make mRNA and stop. But for protein encoding, we have just less than 30 or 32. We have different scientists analysis. So we don't know which one is correct. So if you done that kind of analysis, you can maybe have another different number. So the protein, the dominant protein in our cells is about 50 amino acids to the 1,000 very short or very long amino acids in our body. So each cells have a different set of gene expression, also different sets of protein. So some of them is we have this kind of thing. And that step is we have a filament. This is a kind of filament, very long shape, using for a cytoskeleton in our cells. And also for the seed for where is the seed? This one is kind of seed here. And with also ring, this one a ring. And spur, this one. And also spur like this, different shape. Very diverse. And that's also interesting and very hard to grab from the protein. And you know, it's each species like human, and so you work in human and working on another species, and I work in art and fish, you work in the dog, they slightly different. They slightly different. But totally, in general, they are same structure between the species. They have slightly different. So, okay, if they're diverse. So this slide just shows you they're diverse. And if they are diverse in shape, if you're looking outside, but deeper inside of the protein structure, what is the structure they have it? So normally they have the alpha helix and beta sheet. Because of this kind of organization make the lowest free energy for internal energy of the protein, and they satisfy the bond inside the structure of the protein. So in here is a alpha helix is kind of hard, solid form of protein, and beta sheet is very stable and also flexible. So in here, the keratin protein have a lot of alpha helix there, and like seals, they have the beta sheet. This is the most famous. I don't know. We have another kind of organization form in here, and also alpha helix. They also diverse in organization. Like for some kind of helix, they have just 2 subunits for 1 round, 2 subunit 5 or 6 subunit for one round. And this is just some statement. So many membrane protein cross a lipid layer in the alpha helix. You see that, you recognize it, right? This is a lipid bilayer. This is first layer, second layer, and this is through the layer we have the one protein there. This may be the tail of one protein, and they integrate a fixed protein in the membrane and do the work. You know, is protein is more abundant in the mass compared with the lipid form here. So we have a lot of protein nailed into the membrane to do the work, like receptor or anything, many things. And then a protein is also intertwined alpha helix. So we have the monomer is quite the monomer is an amino acid we know already. So in here is we have the one string of the peptide string, but the structure is somehow in the evolutionary concept. So they can swing together to make this structure is more stable and more harder as a fix for the work. Normally is for the fiber in our cells, like a cytoskeleton or something. And beta sheet is also He also is a lot of structural protein in our cells. And this is they have 2 ways for beta sheet also diverse. Just 2. This is alpha parallels, and this is parallels. That means this is why we have to we can distinguish the 2. This is a N terminal, and this is C terminal, and it connect with the C terminal. And this is a C terminal. No, sorry, N terminal and C terminal in the one arrow in this one. This way is normally we use for beta sheet illustrate. Another question is, in one gene, I mean the same peptide string, sorry. In the same peptide string, they alpha helix and beta sheets co-occur or not, co-present or not? And the answer is yes. They can, in the same string, they have some different kind of domain. The domain is they can one alpha helix, and because another domain is beta sheet is also normal. They belong to the same peptide, also peptide string in one gene. Okay, we have a question.
No question.
Oh, we have a question here. It's a private. Can you explain clear how chaperone guide protein to form protein correctly? I don't know. I don't know about that. So maybe the protein interaction or chaperone is not enzyme. I think not enzyme, not enzyme. So with they just assist the protein folding. I'm not know about the more clear about that. I need to read. So maybe thanks for question. So chaperone in action for protein folding. A longer question. So with the urea and the protein will return the structural native structure if there are no chaperone protein first phase binding or registration. How that the nature protein can form correctly. Like I said that this one protein, how the protein without a chaperone. So the question is without a chaperone, how the protein can return the native structure. So you see that when we increase the urea, and the urea is will destroy the peptide. No, sorry, not destroy the peptide bond. They destroy the weak bond in the protein structure. But when we normally is we reduce the concentration of the protein. No, sorry, concentration of urea. So the bond of the weak bond, like hydrogen bond and ionic bond and vinyl bond, is they reform. And the success of reforming the process is higher in the simpler protein sequence, but more complex. That is not success. That I'm trying already. Without the help from the chaperone. Chaperone just help for the complex. I think it's just help for the complex protein. But in the simple form, we don't need chaperone. We don't need. I tried already. I did. Okay. Okay. I go to the next. This one is. Oh, I don't know. Is we finished this one. But anyway, it's kind of interesting. We talk with protein. So I'm talking another term like a protein domain. Protein domain, like this one. Like this is belong to the protein. So they connect together. They are same gene or different gene. I don't care. But the protein domain is independent in the structure, structural independent pattern of the protein. They can independently folding, and then not connect with this part. So protein need 2 or 3 or more than that to do the work. But domain of the protein is the reason of the amino acid sequence. They can folding independently. So for example, if you are using some molecular technology to cut one domain out of the protein, and you can refolding that protein is refolding domain is more easy compared with you refolding for whole the protein. So and then one domain also maybe do some specific work compared with another one. So one of the very famous example is DNA polymerase. So it's interesting, right? So many people is maybe DNA polymerase is the most studied protein in the world right now, because DNA polymerase for biotechnology and also for disease or anything is very necessary. So The tag DNA polymerase, maybe you heard already. So for DNA copy, right? For DNA copy, in here we have the picture in here. So this is a deoxyribonuclease, okay, RNA. So this is a polymerase. They synthesize another strand of the DNA or RNA from the template. So in the nature, this protein have the two kind of the reaction. This enzyme, they have the two kind of reaction. One is polymerase. I mean, they synthesize the polymer from the monomer. And another, talk a lot. And another domain is they cut something is not correct. Because of they synthesize, maybe the synthesize is not always correct. They detect that one. And another domain they cut something is not correct. That is another word, another function of the protein. And they same protein and the same gene and same structure. So that is the let you know about something about the protein domain. And some kind of protein complex, very big protein, they make from a lot of domain. So and domain 1, domain 2, domain 3, 4, 5 to the number 9 something, for example, they are quite very similar in the structure and also in the sequence. For example, I had an experiment before. I did one. I synthesized one receptor and put into the cells, and that is a membrane protein. That the protein is like the hand, wait for the virus, because I want to produce virus in the cell. For what? For the vaccine. Without a virus, you cannot make a vaccine, right? Because the vaccine is some kind of break structure from the native virus. I need to culture a lot of virus, so I need that kind of receptor for invite virus into the cell, and that receptor have a nice subdomain 1, 2, 3, 4, 5 to the 9, and then the structure is totally same between domain 1, and then they are quite independently. And if they cut this one, and the receptor also work perfectly, no problem. Okay, so go to another one. This one is simple, right? You remember the primary structure like the single string of protein, and secondary the beta sheet and alpha helix for the secondary structure. The third is a tertiary structure. That means alpha helix and beta sheet will be folding a little bit, and then the number 4 is the most complex between the different gene. This gene from this chromosome and this gene from this chromosome, they bind together to form the functional structural protein. This is very beautiful. So we have about 30,000 genes in our genome, in human. And then, but the number of protein is more than that. Because of some gene, they need to combine with another gene to become the functional protein. And using you need some combination, a different combination of the single subunit will make a lot of the protein structure. And in the end of the presentation, I will show you how you can know that information. And you know, we know is that we have the protein structure, we have the beta sheet and also alpha helix. But alpha helix and the beta sheet are quite apart in the solid structure. But the solid is very not flexible to do in the work, right? The protein structure now is very hard and very not kind of flexible to do in the work, because of the protein need to be open for the subject to enter, and it closes, open, close, open, close. Very beautiful. But how is this flexible? We have another kind of thing. We have another type of domain. That is, is an unstructured reason. That means the string. They connect with this domain and they connect with another domain. And then they provide their flexibility for the protein. Very good. And this one also, our lab before using that for the protein making. You know, is when a protein, and if we modify the protein, if we want to modify the protein, the structure of protein is quite a destroyed and not correct anymore. But if you modify the, if you put some kind of protein in the unstructured region, the protein function not change, and the cells still survive well. Cells still survive well. So after that, of course, I am missing something. My protein is very toxic for the cells. If I express toxic protein to the cell will die, right? So I hide that protein inside of this structure, and the cell safe because another structure is covered and protect that one outside for exposed to another part of the cells. The cell's safe, and then I just take it out and cut and take out my protein. That is one trick in the protein making strategy. Okay, now you know it's unstructured reason for the connective. And another thing is, you know, is one, for example, one amino acid string sequence, we have an n number of amino acids, but number of protein in the theory is a lot. Because of each position, we have 20 amino acids. We can exchange 21. But that's why it's not true. Not true. Because we have, you know, nature is very evolution and nature selection very perfect in this thing. Maybe in the beginning, they have a lot of protein making, but none of them is very few of them, very small portion of them have the function. So that only the function one can be favored by nature and then become the function in our living system. So this one is you can grab it, right? Just few of the polypeptide is useful. A lot of them is not useful. And then respect in the DNA sequence in there. So that is if you want to synthesize a new protein, like you put randomly some protein in your protein sequence, if you can synthesize a protein by chemically, and you ask that protein is have the function or not, you have a new structure, a new protein sequence, and normally is not function, I can easily say yes, no function at all. If you try to create a new protein, you have to base on the functional protein and modify it. You have function, but I did try already. I tried a lot. I try modify the protein for a new, maybe it's the sulfur activity of protein. That's in, I make a mutation, but based on some basic protein useful already, and sometimes they got the good result. But if you make from a new thing, it's not work. Okay, next is protein can be classified into the family. That is also from the next slide. Because of the only very limited protein structure can become useful and favored by nature selection. And then the nature selection favor for that. And then they duplicate that kind of case, make to another case inside a single genome. For example, in here we had two enzymes, and you see that they different name, of course, and then also they have a different reaction and different activity. This one is cut for specific, and this is cut for another substrate. But you see that structure quite very same. The green color part is the identical in sequence between the two proteins, and the white color is they quite no function. They have no different sequence. You see that this one is maybe non-structured, unstructured sequence like I mentioned before, and this one is kind of short. This one is long, but the protein structure may be slightly changed. Maybe the active site, the conformation of protein slightly changed, and that make protein is become they also have function, but the subject is different, and make the protein is they have the same structure. No, I mean it's the same structure at some level, and the same 3D structure in total. So this can have a lot of things. One of them is also my research before. I didn't publish this one yet. Another student in Korea is doing it. I'm studying about olfactory receptor. Olfactory receptor is the receptor in our neuron in the nose. It's help we can smell it, and that is the biggest protein family in the genome of the animal. In the human, we have that family, and that family have about more than 900 members, and the structure is totally same. And they just a little bit different in the binding side to the smelling ligand. Okay. Another one is protein from a different they can by chance become the dimer or the tetramer like this, and the structure of this one is totally same between two. This one totally same. And another case is some protein made from slightly different, like beta and alpha. We had this one is hemoglobin protein in our red blood cells. So we have two domain of the beta and two domain of alpha, and that really relate with hemoglobin is the most very clear. About the disease. Study. Hemoglobin gene is a very clear protein target protein for research, because of each mutation of that gene make very serious disease, because of they affect the oxygen transfer ability of the human. In Vietnam, we have a lot of mutations like this. Maybe 20 people, we have 1 people affected with the thalassemia disease. Okay. Another one. And protein can assemble into filament, seed, or sphere. Like this one, they are monomer. And this monomer are quite similar in the structure, totally the same. But they have the binding side. But this one and this one is kind of fit together and make the string is stronger, longer and longer. This is normally happen in the cytoskeleton. You see about when a two set of the chromosome, when our cell is doubling, right, and spindle, right, the chromosome spindle, they recruit this one. So this kind of structure they have a lot. So the chromosome is working in this string to go to another post or go to another post using this structure. It's really beautiful in the cells, and you can see in the lesion of the cytoskeleton. And it's also like this, and it's careful like this. You see, it's the viruses. They have a lot of things like this because of the genome of the virus. Very simple. They just synthesize some very simple protein, and that protein subdomain can connect together, become a capsid, the envelope of the viruses. Okay. And some type of protein have an elongated fibrous shape. Different with this thing, you have a very small and simple form of the monomer like this. In this case, we have a very long monomer, and then some monomer they can tangle together to make a complex and a stronger structure in the cells. And this is showing you the covalent bond between the protein and protein. This normally helps the protein can bind together because of you see that this is cysteine, right? Cysteine, very covalent, very stable in the cells. This is for example, the peptide 1 is they have the alpha helix bond, and then they localize into the cell surface. But this one is not localized in the cell surface, but inside of the cells or outside of the cell, this one. But they can connect with the membrane protein and become the covalent bond. And then they, this bond is hard to break in the cells. You have to use very strong detergent to break the cell and release this one and this one because covalent. If you have the hydrogen bond or some kind of weak bond, we can easily to break using some heat, using some pH change in a pH, acid. So we can, in the acid, you can release the protein structure in the cells easy. I did try already with about 4 or 3 pH. The protein membrane will be removed a lot. Okay. Okay, any question about the first part, the structure of protein? I think it's quite clear, right? Nothing. We just go to a little bit deeper about the protein organization. Okay, I will go with this part fast, how protein work. So protein work in case of they can bind to another molecules. That molecules is maybe the protein or another ligand or another simple chemical in the cells. Maybe the simple molecule signal in the cells. And you can see it's very diverse in later. They have to be a physical interaction. They cannot telephone or something. This protein talk to protein by telegraph or something. No, they have to physical interaction. That's it true. And each protein can bind just one or few molecules out of many thousands of different molecules. I mean the specificity of protein. This protein was born for specific reaction. But we have some exception. Example I will show in some few slides later. Like, and that specificity can show by antibody and by a virus, a vaccine. And some enzyme, you know, it's the enzyme named by the substrate because of the lock and the key the interaction between the two. So this one I've shown already, right, from the last slide, last presentation. So we have the ligand here and the binding size, and we have a lot of binding, weak binding between two. So what a problem if we have a covalent bond between the ligand and the binding site, for example. So because of ligand and the enzyme protein is quite a weak, right? Because sum of the many weak bonds between two is maybe not stable because of maybe just 1 minute they will disassociate, and stronger associate maybe have a function, but the weak they will easy to release and no any kind of reaction happen because very fast. So can we apply the covalent binding here? Oh, okay, okay, just go. So I upload the video after this. Thank you. This is a visualize for this is protein structure, and this is active site. We have a lot of side chains of the protein, and this is ligand. So they fit each other by very subtle binding, right? So each side of mutation in here will change the binding, and then the reaction is not happen with 100% like nature anymore. They can increase or they can release. But any changes may be harmful for the cells, may be harmful for the cell. And that is the basic of the disease. For example, if I modify that kind of glutamic acid in here, and this molecule cannot bind to this protein anymore, and that biochemical pathway cannot work, and the cell has a problem. But due to that pathway important or not. So single mutation on the genome and make a protein change, and the reaction cannot happen. That's basic of the disease in the cells. So the binding between the protein and the ligand is a very important thing. And the antibody example is you already know about that, right? So it's very diverse. A lot of bacteria, a lot of ligand can bind to very specific to the only one antibody. And our cells is produce a lot of antibody in our life. You see, its antibody kind of very stable. So I remember last time. Okay, yeah, right. No covalent, I forgot. No covalent can be. So if they bind very well to the protein, and you know its enzyme have to be finished this reaction and releases and come to another substrate to do it. If the covalent, they cannot release that kind of enzyme, they cannot release that kind of substrate, and the enzyme is die, right? So no covalent is favored in the selection. Talking about the stable of when I was first time working on with antibody very expensive. So I'm worried about the stabilized, stable form of the antibody. But antibody very stable because they have a lot of cysteine here. You see, SS is covalent bond. So I can send to you as for my friend to help me to do experiment because of I have his have the good skill and I have the antibiotic. I just send it during the airplane. So antibody very stable. See, a lot of covalent bond here. So this one is in immunology class you will learn already, right? So antibody is very specific for the ligand. The ligand may become foreign molecules or the viruses or bacteria. If you want, we can discuss about a very beautiful story about the antibody. Remember, antibody is one antibody can bind to one type of ligand only. Sometimes we have that kind of contamination. I mean, it's cross linking, cross reactivity. I mean, this antibody is not well produced, so they can bind to another two types of the molecules. That's also happen. That's why in the COVID-19, the pandemic now we have suffered. We don't use the antibody technology for detect the virus because sometimes it's not correct because we have a lot of coronavirus, right? So we have to use another method, and now we use RNA detection. So some of you is learning about PCR something, right? Okay. So oh, we have some slide more. Just some slide more, and we can finish this chapter. You know, it's one protein will bond for specific reaction with another protein or another substrate. That specificity of the protein binding very high. But I have another, due to my research, my study before in my PhD, I have tested with this one. I'm working on MXC. So MXC is a different case. So MXC is a major histocompatibility complex. So it is cell surface protein, and they can bind one molecules. They can bind with a lot of ligand, maybe million ligand, and based on the high binding or low binding, we have the reaction, the immune reaction. This molecules is one major immune reaction in your body. So your body is have one set of this kind of molecules, and that's very important. That becomes of your native thing. Because you know what? You see, some people can resistant with some disease. Maybe some people can resist with some people is not in fact with the with what? With HIV, or some people is resist with some bacteria thing. But maybe you is not good. You will suffer a lot. Some people is not, because the immune system. These molecules is can by I have the set of these molecules and can react well with this bacteria, and you have another set of this bacteria, this set of the molecules MXC. So you can resist with another types of bacteria. So so I want to mention here, because this is quite a receptor, not specific a lot. They can bind with many types of their ligand, and that is the nature. My lab also working in this, my lab in IU also working in this kind of molecules. I mean, in the gene level, not protein level. And this table show many types of protein class. So with a different specific function in the cells. We have ligase, we have protease, nucleus, many. You can check in the book. And another one is lysosome. Lysosome is an enzyme used for the cut the sugar cell wall of the bacteria. You know, the bacteria is not like bacteria cell membrane is not simple like human cells. They have the cell wall. Bacteria have a cell wall. If you break the cell wall, bacteria will die because they don't have their cytoskeleton. Cytoskeleton only in the human cells. In the plant cells, the cytoskeleton also very less. They also have cytoskeleton using for the cell doubling, like DNA duplication. But because of the plant cell, they have cell wall already. Same with the bacteria, so they don't need it. But human don't have the cell wall. So lysosome is an enzyme in the saliva, tear, or other. They become the one factor, one thing of the native immune system, different with adaptation immune system. So they can cut the sugar cell wall bacteria make by the glucose. So this enzyme will cut very precisely. They cut glucose and fructose and break it, and make the bacteria is die because of the cell wall break it. So in here they illustrate for that kind of activity. So we have the ligase, the substrates in here, and this is some amino acids like 35 by glutamine and opposite acids in the 52. Okay, and in here in the bone, so you know, it's the reaction in a biochemical reaction, also chemical reaction relate with the electron transfer. The electron in here we transfer to this one, but the structure is same, right? Just electron transfer, and the proton like the hydrogen proton is out. So this one transfer to oxygen in here. So oxygen now is have the electron already, so they don't need electron from the C anymore. So they break in here. So that is kind of the mechanism of the enzyme using that one. So enzyme can donate electron because they're very close. So they're easy to donate electron from here to here, and they can easily revert. They also have it back from where? From the water outside. So proton is the water is have a lot. So, and this is also another mechanism of the protein. enzyme activity. So they provide accommodation, position for the two ligand can enter, and they charge or the what, and changing the protein conformation. And this also important. Many drugs inhibit enzyme. Many drug inhibit enzyme. So what drug is kind of protein, or drug can be the organic chemical or inorganic chemical, normally organic chemical. So they can inhibit the enzyme by they can bind to the active site of the enzyme, or they can compete with the substrate of the enzyme, or somehow they can bind to the allosteric site of enzyme. It means that the enzyme in here, this is an active site, and this is another site for regulation. So the drug can bind here to make the conformation of enzyme change, and enzyme have no function at all. Some drug is also include the activate enzyme. What is drug? That is vitamin, right? Vitamin is a cofactor of enzyme. They can, when we use a vitamin, and enzyme activity will increase. Also drug. So some enzyme is inhibits some very basic pathway of the gene in the cells, make the cells cannot propagation anymore, cannot doubling anymore. So that drug using for cancer, because of cancer, the cell is propagation uncontrolled. So the drug treat the cells, and the cell cannot produce anymore. So we say. And another drug is lower used for the high level people, cholesterol, high cholesterol people. So they inhibit some enzyme making the cholesterol in the cells. And another thing is some molecules need for extra function of protein, because of the like hemoglobin cannot eat cells storing the oxygen in the cells. So they need a HEM. This is what we call the HEM molecule. This is structure of HEM, and this molecule is the center of the which one? This one. HEM should be in here. This is HEM, red one. So HEM can connect with the oxygen. Protein itself cannot connect with oxygen. So they need that for the function. Okay. Okay, maybe time is not enough now, so we can stop today. So the remaining part, maybe you read it, or I can provide a video about this one. I can provide some small story about the protein, and for you can a little bit deeper understanding of protein, how protein control, and another one is how protein is studied. It's very interesting because it's that related with biotechnology, and also is medical research. So thank you very much. So do you have any question? Question. Any question? Hello? Connection correct? Okay, so you can post to the Blackboard. I will open the Blackboard section discussion for this part in the Blackboard. Just put the question there, and I may also have some kind of report to you. I mean, the assignment. So I'm also thinking about midterm question. Some question. Okay, thank you very much for joining today. I will submit. I will post the PPT file and also the video file for anyone cannot attend today. And thank you very much. Thank you. Okay. Okay, goodbye. Have a nice day. Stay safe. Thank you a lot. See you next week. Hope is we see each other physically soon. Okay, bye.